If, like me, you've been trying to improve soil health in your garden or on your farm, then you'll have been doing things like cover cropping, making compost, spraying compost teas, broad forking, maybe using some worm compost. Then we need to find a way to monitor the progress that all of these amendments are making to our soil and our plant health. So in today's video, I want to go through the various ways in which I monitor soil and plant health here on my flower farm. And I'm going to go through a list of things that we can do for free or that are relatively inexpensive, through to the more expensive monitoring techniques that we can use on our farms and in our gardens to make sure that we are moving soil health in a positive direction, and what we're going to be doing is regenerating our soil rather than just being sustainable or even degrading our soils. Healthy soil is the backbone to a healthy ecosystem overall. And those healthy soils are going to be full of beneficial microbes that are going to create a great environment for plants living within it. And those healthy plants are going to be more disease resistant, more pest resistant, and they are going to be more nutritious for us, healthier. So if we're producing flowers, they're not going to have signs of disease or pest damage. So in order to find out whether we're moving towards those healthy soils and healthy plants, we need to find monitoring techniques to make sure we're going in the right direction. And these are the techniques that I use here on my farm. So I think that one of the best free tests you can do to test soil health is to do a worm count. And worms are beneficial in so many ways in the soil, so the more the merrier in soils. And the way to test them is to dig a hole. You want a standardised size hole each time you're doing this, so you can keep a record of how it changes over time. And actually now isn't really the best time to be doing worm soil counts because it is pretty dry here. We're in the middle of summer. The best time to be doing it is early spring or when the soils start getting a bit damper in autumn. But I'm going to do it now anyway for demonstration purposes, and I'm going to dig a hole roughly 20 centimetres cubed. So I'm going to do it 20 centimetres deep, wide, long, and we're going to remove that material from the hole that we've dug and put it into a bucket, and then we can go through that sample and count the worms that are in it. Okay, so the moment of truth. I need to put these worms back because they're not looking very happy. There's more than I thought actually. We had 16 worms overall. The size of the hole was basically the width depth of this spade, which is just over 20 centimetres, probably more like 25. So I would be interested to know in the comments, if you could give this test a go, how many worms do you have in your 25 centimetre cubed piece of soil? The next test that we can do, which is free to do, is the Slate Test. And this test tells us about the aggregate stability in clumps of soil. Soil that is healthy and has a good microbiome will have macro and micro aggregates, which are small pore spaces in the soil which are glued together by glomalin and glues that the bacteria and organisms within the soil are releasing, and that just helps to create a stable and kind of constant supply of air and water within the soil structure, and means that we don't get waterlogged and we don't get soil erosion. So if we haven't got those microbes present, then the soil very easily erodes away and can become waterlogged, and we don't have those air pore spaces in the soil. So in the Slate test, we are going to be putting a chunk of soil in water to see how it reacts. A healthy chunk of soil with lots of aggregates and aggregate stability will hold together and the water will stay relatively clear, whereas degraded soil will kind of break down because there isn't any of those microbial glues holding the aggregates together, and we will get turbid water and the soil will just break apart and dissolve into the water. So I've got a sample here from my field that's had 2 cover crops on it. This is the second cover crop of the season. So I'm hoping that this is going to be some good soil that's going to have some good aggregation within it. I can already see, kind of just turning it over and looking at it, that it has got some pore spaces that maybe have been created by some larger organisms within the soil food web. So I'm hoping that this is going to stay together quite nicely in the water. So basically what we're going to do here is have a vessel of water where I can see quite clearly in the water column there, and then I'm going to put a sieve on the top of the water. I might just top up the water a little bit once the soil is in there, and then I'm going to put my piece of soil in the sieve there and just watch what happens. So the slate test has been going ahead for a few minutes now, and the sediment or the kind of breakdown of this clump of soil seems to have kind of stopped now. There was some initial kind of sprinkling of some soil that's fallen down at the bottom of the jar, but now it's kind of settled down. I need to get this thing to stop looking at me. And as you can see, this piece of soil has kind of stayed in its original shape, which shows that the aggregates are quite stable within this clump of soil, which is good to see. If the aggregates weren't stable, then you would maybe notice that this piece of soil would start slumping down. What I might do actually is get some soil out of the agricultural fields, the fields that get worked every season, and just see how that performs in comparison to this, because obviously these two soils will be managed differently. So I'm quite pleased with that. I've just disturbed it, so there's a little bit more soil falling down there, but it seems to be relatively stable. I will keep this in here for a bit longer just to see after a longer period of time how that soil reacts, but overall that looks like a positive result for the slate test. Another thing that we can do that is completely free is to be able to just make observations. So observing the soil, how does it look? Is it looking quite nice and loamy and dark coloured, which is the sign of a good soil with high organic matter? Does it have a crust on top? Is it cracking open? Is it bare? We don't want bare soil. Remember, we want our soil to be covered at all times. So that's why I use either the plants that I'm growing as a crop or a cover crop to try and keep living roots in the soil at all times. And also you want to look at the kind of weeds that are present within the soils. So weeds are a really good indicator of what is going on within your soils, and there are some good resources out there online, of course, and I've got this book which seems to be quite handy just to find out the kind of role of weeds in the ecosystem. And up until a couple of years ago, I thought that weeds were the enemy, and every time I had to kind of go on a weeding sesh, it used to make me really frustrated, and I used to get quite angry with the weeds. But nowadays, I see the weeds as kind of something that is just functioning within the ecosystem. They're trying to progress the ecological succession of the area by providing some kind of ecosystem function. So if we think about compacted soils, we might see things like docks and thistles, and they tend to have really deep tap roots which are going to help to relieve compaction within the soil. So once those dock leaves have done their work, then they are typically not going to get the signal to grow in that area, because that area has moved beyond the stage of succession where they are useful within the ecosystem. I hope that makes sense. So you might just want to have a look around at what type of weeds you've got, research what their function is within the ecosystem, and it might give you a better idea of kind of what's going on, what kind of ecological stage of succession you're at. So remember that we go from kind of bare areas, we've got those early colonising plants which are going to kind of help to add organic matter and improve the conditions for the next succession. And you might remember those diagrams from science. I'll put one on the screen now, and we go from kind of bare land up until kind of an established forest. So you might see, as you start to build more fungi, more of a microbial healthy soil, you might start to see kind of more woody, shrubby kind of weeds germinating in your fields, which are indicators of a later successional stage in kind of the ecology of the area. So and those are just some things to look out for. I'm not going to go too deep into that, so you can do your own research into that if you like. The other thing we can keep an eye on as well is the kind of pests and diseases that our plants have. And healthier soils typically have healthier plants, obviously, and really healthy plants, which we're going to go on to kind of how we can assess plant health afterwards with this Brix Refractometer, which costs a little bit of money, but I think it's worth it just to be able to kind of monitor how our plants are doing. But healthy plants cannot be detected by insect pests. So the more kind of we can put into soil health, the more the plant health develops, and that is going to help our plants be undetectable by insect pests. But we are going to go on to that next. So we're moving on now to the kind of less expensive soil health testing methods that we can use to kind of assess soil and plant health on our farms and in our gardens. And this one I really love. It's the Brix Refractometer, and we use this to kind of test dissolved sugars and minerals within plant leaves. So what we do with this is we take some leaves from the plant that we are interested in looking at. So I've got some leaves here from the cover crop that is in one of my beds at the moment, so I'll be interested to see what the Brix levels are in these leaves. So what I'm going to do is just crush them up, and then I'm going to squeeze out the juice, take some into a pipette and squeeze that onto, oops, and then what we do is we look towards the light, and I would say that that is reading about 7.5. So for a healthy plant that is not going to be detectable by insect pests, we want the Brix level to be kind of around 12 or above. So it could be that today it's quite early in the morning, the sun, it's only 9:30. So I'm thinking that maybe we'll try it again at midday, because the plant hasn't been photosynthesizing for very long. So we'll test it again later and see what happens. It might just be that the Brix level is 7, but we can check again later on in the day. It's good to kind of test at different times of the day just to see how the plant is responding. And also it's good to test it after you have sprayed amendments in the soil, so that you can see whether those amendments are helping to increase photosynthetic rate. So the way to increase Brix level is to increase photosynthetic rate, and that is kind of the $1 million question. How do you do that? So you want to just create really healthy soil, make sure the plant's got everything that it needs, and you are going to, as a consequence, have a healthy plant. So you kind of just, it's a long-term goal. You're not going to put something on and the next day the Brix level is going to be above 12. It's kind of something that we're all striving towards, is having healthier soils and plants that are resistant to pests and diseases. I've got some more leaves here that I've just collected from the field, and those are from my snapdragons, so it's going to be interesting to see how healthy the snapdragons are. Okay, so snapdragon Brix is very good. It is probably around the 14 mark. So chuffed with that. That means the snapdragons are very healthy and the pests aren't going to be able to detect those snapdragons. So keep your hands off pests. So those snapdragons were from an area of my flower field where I've added lots of organic matter. A couple of years ago I added loads and loads of cow muck onto it, and I covered it over with a sheet and I left it for a full season. When I peeled the cover back, the worms were just going crazy, and the whole surface of the soil was just like a huge wormery, and it was just full of beautiful worm castings, and I was really pleased with how the soil looked. And since then I've set up some no dig beds, I've added more compost on top. The paths are made of straw, which is also adding to the organic matter, and those beds just seem really healthy to me. So what I'm going to do actually is I've got another bed of snapdragons planted out in the main area of my field, which has probably had less organic matter added to it, kind of probably less microbial activity going on there. So I would be interested to see how those snapdragons are faring compared to the ones that are planted in the area that's got high levels of organic matter and kind of just seems to be doing well. So I'm going to go grab a sample and come back and see what it looks like. Okay, so I would say for this sample of snapdragons that the Brix is probably around 11 or 12. It's quite difficult to tell with this because it's more of a gradient than a line, which indicates the mark of Brix. So an unclear line where you are taking your reading from may indicate that you have quite a lot of dissolved minerals and nutrients within the sample. And if you have a very dead sharp line, that might indicate a calcium deficiency. And I can't remember what sample I was looking at actually, but a few weeks ago I did notice a very, very sharp line between kind of when you're looking down the refractometer, you can see there's a yellow section at the bottom and a blue section at the top, and it was a very, very dead straight line. But looking at these snapdragons, it's more of a gradient from yellow to blue rather than kind of a very clear indication of what the Brix levels are in the plant. The Brix levels typically peak in the afternoon, and as I said earlier on, it's kind of 10 o'clock in the morning here, so it might be worth checking them again later on in the day just to see how it changes throughout the day. And also it's a very sunny day today, which is a good time to test because the plants are photosynthesizing nicely. The other thing to test is the weeds that you have growing in your field or garden. If your weeds have a higher Brix level than the plants you are growing, then the area is kind of more suited to the weeds, and you need to kind of do something about that to make sure that the plants that you've planted out are going to be able to outcompete the weeds. But if the weeds are photosynthesizing more efficiently than the plants that are in your garden or in your field, then they are going to outcompete your desired plants, and that's not going to be the outcome that you want. Another low cost test that you can do, which is quite interesting to have a look at, is organic matter. And most soils that have been kind of worked over time, maybe ploughed, rotavated, dug over, are going to be a little bit lower in organic matter. And what we're going to be trying to do if we're working on soil health is to build that organic matter in our soil. So if we've been adding cover crops and we've been keeping our soil planted with living roots at all times, then we will be building organic matter in our soils. So what you can do is quite simply send off a soil sample to a local lab that is doing soil testing, and you can have your percentage of organic matter tested as a benchmark to, if you've just started on your soil health journey, you can see how soil organic matter improves over time. And if you are kind of midway through your journey, there's no harm in doing it now. Although maybe you already have done your soil organic matter testing. I would love to know what results you got down in the comments below. I recently had my new flower field tested for soil organic matter because I wanted to know where I was starting with it, and because this field is going to be kind of managed with soil health in mind from the very beginning, I just wanted to know what the soil organic matter content was. But anyway, the field that I have just converted into a flower field, all I've done with it so far is killed off the grass and grown a cover crop on it. That field was soil organic matter content of about 14%, I think, which is absolutely amazing actually. It has been a long-term grass paddock with old grasses on it, so it's not surprising that it has high organic matter. And then my other flower field, which is the second flower field that I set up where the polytunnel is, that had about 12% organic matter, so that's a really good number as well. Most agricultural soils have got a soil organic matter content of about 1 to 2%. So that is kind of what we're looking at there. So yeah, it's interesting to see how soil organic matter can build over the years. You might be able to add a percent or two of soil organic matter every year if you're just constantly cover cropping it and managing it in a good way. So it's going to be interesting to see how we can get that creeping up over time. Next up, we are getting into the slightly more expensive soil analysis tools. And here I have a Microbiometer which Rob and I purchased from Groundswell this year. I've got Rob to blame for this really, because he likes a good gadget, so he fancied buying one of these and now we have one. So this tool helps you to analyse the microbial content of your soil and also the fungal to bacterial ratio. We're aiming towards building more fungi usually in the soil, because mycorrhizal fungi help to kind of extend the reach of the plant's roots, and they work in symbiosis with the plant to be able to bring it nutrients that the plant wouldn't otherwise be able to reach within the soil. And if we are kind of tilling the soil or rotavating, digging, then we usually break those mycorrhizal fungi strands, and the fungi just isn't able to function properly in the soil. It's kind of much more delicate than bacteria and other organisms within the soil. So mechanically disturbing them really sets them back, and they cannot thrive in an area that is being constantly disturbed. So through no-dig practices, we are trying to encourage fungi within the soil. We might want to add fungally dominant composts like the Johnson Sioux bioreactor composts. We might want to add wood chips to encourage fungi within the soil, which is going to help create those symbiotic relationships with plants. And this microbiometer will help us to assess the fungal to bacterial ratio in the soils. So from kind of what I've read and what I've listened to online, that a 1-to-1 fungal to bacterial ratio is a good ballpark figure to aim for, because there's a balance of bacteria and fungi, and the plant can use those to its own benefit and can choose who it wants to kind of get on its team. And actually what I would recommend is reading these books by Jeff Lowenfels. I have a teaming with fungi one somewhere. I don't know if it's in here. So I've got teaming with microbes, teaming with fungi. I think he's got a teaming with bacteria and a teaming with nutrients, I want to say. I'm not quite sure, but I'll put the links down in the description to those books. These books just give you all the information that you need to know about how plants work with fungi and bacteria to get what they need from the soil. So you have to bear with me on this because I've only used this once, but I'm going to I've taken a sample from the area that I took my initial Snapdragon leaf sample from. So I'm thinking that this might have a high fungal content. I'm not sure, we'll see what the test says. Here I am on the Microbiometer app, and what I'm testing here is soil. You can also test compost, compost tea and compost extracts. It would actually be interesting to have a look at the compost pile that I have just finished and turned to see whether that's got a higher fungal content in it. And then we've got you can make it part of a project if you want to. So I am going to start the test from the beginning because I can't remember exactly how to do it. Once I've done it a few times, I can just use this card and test out my soil on there. The fungal to bacterial ratio here is 1.3 to 1, which is kind of what we want. So that's looking good. We've got a high microbial content, getting on the high end of a fungal to bacterial ratio. So yeah, looking good. So I just wanted to mention that this kit, I can't actually remember the price of it. It was quite expensive. I think it was 150 quid or something like that. And then I think it's about £10 a test. if you want to buy the refills for it. So obviously that's a bit of an investment. Maybe you don't want to spend that much money on testing your soil, but you can get this done in a lab as well, and I think that soil smiths do it. They definitely used to do it. I'm not sure if they still do, but I presume so. And you can get that fungal to bacterial ratio done in a lab like that, so I'll put a link down to them, and you also might get a bit of a feedback from them as well, from the lab, saying whether it's kind of good or bad, and what kind of management strategies you might want to go on to next to try and improve that. And the final, and probably one of the most expensive ways to test soil health, is with a microscope. And this microscope did set me back a fair amount, but you guys helped me to pay for it, because I received lots of buy me a coffee donations at the time when I was looking to purchase this microscope. So I just have to say, every time I use this microscope, thank you guys, because you helped me to come on this soil health journey. And I'm trying to give back to you guys by showing you microscope footage and kind of providing the information that I'm providing on videos like this one. So the sample of soil that I took from the bed that was showed the nice results on the Microbiometer, I have some of that soil here, so I'm going to have a look at it under the microscope as well, because the Microbiometer tells us the fungal to bacterial ratio, which can also be counted on the microscope, but I'm not quite qualified to do those quantitative analysis on the microscope. So we can just kind of get a visual representation of what's going on in the soil, and we can look at those beneficial microbes. We can even look to see if there's non-beneficial microbes, or that if our soil's going anaerobic, or if we've got kind of like an imbalance of organisms in our soils. We can look for nematodes that might not be the right kind of nematodes that we want. We don't want root feeding nematodes, for example. We want beneficial ones that are eating bacteria and kind of forming a part of the soil food web. So by getting a visual representation through the microscope, we can kind of look to see whether things are heading in the right direction. So I'm going to pop these samples on a slide and we can take a look at what's there to see. So when I first pop a slide on the microscope, I'm looking at 40 times magnification, and I can just have a scan through this slide and see whether I can see any of the larger organisms like the nematodes. So I'll usually scan through, see if I can see any nematodes, maybe give them a quick count up, and then I will zoom in a little bit further to see what I can see at a higher magnification. Once I'm zoomed in, I am working at 1,000 times magnification, and I'm looking for smaller microorganisms like bacteria, protozoans, and fungal hyphae. And you can see in the left-hand side of this image here, there's a protozoan, a testate amoeba, and they feed on bacteria. So we can also see varieties of different types of bacteria. So we can usually distinguish bacteria by the shapes of their bodies. Protozoans are a little bit bigger. If we were looking at anaerobic conditions here, we'd be seeing ciliates, but we're seeing mostly aerobic conditions here. And I'm just looking for a nice variety and a nice mixture of all the different organisms in the soil food web to make sure that the sample is looking healthy. Thanks so much for taking the time to watch this video about soil health monitoring and plant health monitoring, and I hope you found it interesting and informative. If you have any questions, pop them down in the comments below and I'll try my best to get back to them. And if you have any observations that you've made that you would like to share with us down in the comments, then please do that as well. Thanks again, and I hope to see you again soon.